Hello everyone, myself Shreya Paliwal, faculty Zoology in Resonance, pre-medical division. So we are here to discuss the NEET 2019 solution which was rescheduled for the Odisha. So let's discuss the paper now. Question number 45. Which of the following receptors are specifically responsible for maintenance of balance of body and posture? This is the direct question from the NCERT from the chapter Neural Control and Coordination. The first option says basilar membrane and otoliths. Basilar membrane is actually the part of the cochlea. In our inner ear, there is one vestibular apparatus consist of utricles, saccules and semicircular canal. So, these otoliths are the calcium carbonate particles which are present in the utricles and saccules. So, these are responsible for the maintenance of the body, they sense the body position change, but basilar membrane is the part of cochlea, so it is basically involved in the hearing. So, option number 1 is wrong. Again, the option number 2 is having the organ of Corti, which is a hearing organ, so this is also not the correct option. Option number 3 is tectorial membrane and macula. Tectorial membrane is again the part of the cochlea, and macula is actually the sensing part present in the inner ear. So, it actually responsible for the maintenance of body equilibrium in the static position. Now, we are having option number 4, that is crista ampullaris and macula. Crista ampullaris is actually when we talk about the semicircular canal. So, in semicircular canal, the base part is swollen and that is known as crista or crista ampullaris. So, crista ampullaris is responsible for maintaining the body position or body equilibrium at the time of movement, and macula is responsible for the body equilibrium maintenance at the time of static position. So, option number 4 is the correct option, and in NCERT directly the line is given that crista ampullaris and macula are the specific receptors for the maintenance of balance of body and posture. Question number 60. Which of the following statements is correct about the origin and evolution of man? This is again the direct NCERT based question. The direct lines are given in the NCERT in the evolution chapter. First option is agriculture came around 50,000 years back. If you read NCERT, then it is clearly given that agriculture came around 10,000 years back. So, this is the wrong option. Option number 2, the Dryopithecus and Ramapithecus primates existing 15 million years ago walked like men. So, this 15 million years ago is the right part, but they were not actually walked like a man, they walked like a chimpanzee and gorillas. So, this part is wrong. So, option number 2 is also wrong. Option number 3 is Homo habilis probably ate meat. No, Homo habilis probably didn't eat meat. So, this option is also wrong. Option number 4 says Neanderthal man lived in Asia between 1 lakh and 40,000 years back. So, this line is clearly given in the NCERT. So, correct option will be option number 4. Question number 63. Prostatic groups differ from coenzymes in that. This is the straightforward question from the biomolecules chapter. In cofactors, we are having 3 types: prostatic group, coenzymes and metal ions. So, the option number 1 says they require metal ions for their activity. No, it is not the correct option for both of them. Then second option says prostatic groups are tightly bound to the apoenzyme. This is the correct option, as prostatic groups are the organic compounds and they tightly bound with the proteinaceous part of the enzyme, that is apoenzyme. Option number 3 is their association with apoenzymes is transient. This third option is correct for the coenzymes. Coenzymes are also organic compound, but their binding with the apoenzyme is transient, usually occurs when during the catalysis reaction. So, option number 4 is they can serve as cofactors in a number of enzyme catalyzed reaction. Again, this option is wrong. So, for the type of cofactors, prostatic groups and coenzymes, the correct option for the prostatic group is they are tightly bound to the proteinaceous part, that is apoenzymes. Question number 65, Ramchandran plot is used to confirm the structure of. This question is in, if you see the NCERT of class 11th, the chapter 8, Cell the Unit of Life, in front of that chapter there is a page given in which scientist, there is a description of a scientist G.N. Ramchandran. So there one line is given about the Ramchandran plot as the G.N. Ramchandran actually worked on the structural biology part. So if you see the options, first is RNA, that is wrong, second is proteins, third is triglycerides and fourth is DNA. Basically जो रामचंद्रन प्लॉट होता है वो कंफर्मेशन जो स्ट्रक्चर चेंज होते हैं प्रोटीन्स के लिए उसके लिए बेस्ड एक पर्टिकुलर पार्ट है. सो प्रोटीन्स में जो NC बॉन्ड प्रेजेंट होता है बिटवीन द टू अमीनो एसिड, देयर इज अ NC बॉन्ड. उस NC बॉन्ड के एंगल रोटेशन की वजह से जो कन्फर्मेशन आते हैं दैट इज एक्चुअली shown by the Ramchandran plot in a square form. different areas होते हैं, different areas में वहाँ पर कुछ structures present होंगे कि ये particular structures वहीं पे अपना confirmation दिखाएंगे, like phi bond होगा, psi bond होगा. So that is very detailed part, but in NCERT there is a one line in that particular paragraph about the scientist. So the correct option will be proteins. Question number 77, a selectable marker is used to. This question is from NCERT from the chapter Biotechnology. The options are, first says help in eliminating the non-transformants so that transformants can be regenerated. Yes, this option is a correct option. If you talk about the transformation means जब भी कोई आपका host cell foreign DNA को uptake करता है from the surrounding environment तो उस process को हम transformation बोलते हैं. So basically अगर किसी host cell में for example E. coli most common example है, अगर उस host cell ने foreign DNA को uptake किया है so that will be known as transformant. और अगर किसी होस्ट सेल ने फॉरेन डीएनए को अपटेक नहीं करा है तो दैट कॉलोनी विल बी नोन एज नॉन ट्रांसफॉर्मेंट. सो बेसिकली जब भी हम क्लोनिंग वैक्टर की बात करेंगे तो वैक्टर में एक प्रॉपर्टी होती है दैट इट मस्ट हैव अ सेलेक्टेबल मार्कर. सेलेक्टेबल मार्कर आर ऑफ मैनी टाइप्स. फॉर एग्जांपल मोस्ट कॉमन इज एंटीबायोटिक रेजिस्टेंस जीन, फॉर एग्जांपल एम्पिसिलिन रेजिस्टेंस जीन और टेट्रासाइक्लिन रेजिस्टेंस जीन. सो बेसिकली क्या होगा कि अगर आपने क्लोनिंग वैक्टर में आपकी फॉरेन जीन या जीन ऑफ इंटरेस्ट को इंसर्ट किया है एंड दैट वैक्टर इज ऑल्सो हैविंग द सेलेक्टेबल मार्कर, फॉर एग्जाम्पल एम्पिसिलिन रेजिस्टेंट. सो जब ये क्लोनिंग वैक्टर आपके होस्ट सेल में एंटर करेगा सो दैट बिकॉज ऑफ दैट वैक्टर इज हैविंग द एम्पिसिलिन रेजिस्टेंस जीन, वो पर्टिकुलर चीज़ पहली बात तो ट्रांसफॉर्मेंट होगी. अब अगर मैं उसे किसी प्लेट पे ग्रो करूँ उस पर्टिकुलर ट्रांसफॉर्म्ड कॉलोनीज़ को दैट इज़ हैविंग द एम्पिसिलिन, सो एम्पिसिलिन की प्रेजेंस में भी वो पर्टिकुलर बैक्टीरियल कॉलोनी ग्रो कर जाएगी बिकॉज उसके पास एम्पिसिलिन रेजिस्टेंस जीन प्रेजेंट है. ऑन द अदर हैंड अगर किसी होस्ट सेल ने पर्टिकुलरली नहीं लिया है फॉरेन जीन को या क्लोनिंग वैक्टर को, फॉरेन जीन इंसर्ट नहीं हुई थी और सेल ने उसको अपटेक नहीं किया है, सो दैट कॉलोनी विल बी नॉन ट्रांसफॉर्मेंट. सो ट्रांसफॉर्मेंट और नॉन ट्रांसफॉर्मेंट को हम बेसिकली सेलेक्टेबल मार्कर के बेसिस पर डिफ्रेंशिएट कर सकते हैं बिकॉज दोज आर हैविंग द एम्पिसिलिन रेजिस्टेंस जीन, वो तो ग्रो कर जाएंगी easily on the plate, बट जिनके पास एम्पिसिलिन रेजिस्टेंस जीन नहीं है वो वहाँ पे ग्रो नहीं कर पाएंगी. सो ट्रांसफॉर्मेंट कॉलोनीज़ को फिर हम फर्दर लेके हमारा वर्क ऑफ इंटरेस्ट कैरी फॉरवर्ड कर सकते हैं. सो द करेक्ट ऑप्शन विल बी ऑप्शन नंबर 1. क्वेश्चन नंबर 84, which of the following statements about methanogens is not correct? Methanogens belongs to the domain Archaebacteria. So the options, let's see the options. First, they can be used to produce biogas. Yes, this is the correct option. It is given in the chapter Microbes in Human Welfare. Then option number 2, they are found in the rumen of cattle and their excreta. So this is again the correct option. they grow aerobically and break down cellulose rich food. We know that methanogens are anaerobic bacteria, so this aerobically term is wrong. So option number 3rd is actually the incorrect option, and the option number 4 says they produce methane gas. So the production of the methane gas is also the function of the methanogens, so this is again the correct option. So option number 3 is incorrect. Question number 86, coca alkaloid or cocaine is obtained from. This is a straightforward question from the NCERT. The chapter is Human Health and Disease. Drugs wale part mein this line is given that coca alkaloid or cocaine is obtained from the erythroxylum coca. So you don't have to think much. It is a clear cut line. There is a clear cut line in the NCERT. So the correct option will be 3. Question number 87, among the following pairs of microbes, which pair has both the microbes that can be used as biofertilizers? Biofertilizers are basically, they actually add the nutrients in the soil. So we know that rhizobium bacteria fixes the atmospheric nitrogen, and cyanobacteria, some cyanobacteria also fix the atmospheric nitrogen, hence they are increasing the quality of the soil, and some cyanobacteria or blue-green algae are adding the organic matter to the soil. So the correct option will be 3rd. This question is directly given in the NCERT in the chapter Microbes in Human Welfare. Question number 88, given below are 4 statements pertaining to separation of DNA fragments using gel electrophoresis, and you have to identify the incorrect statement. Let's see the options. Option number 1 says DNA is negatively charged molecule and so it is loaded on gel towards the anode terminal. DNA is negatively charged and anode is positive electrode. So you can't load any sample of a DNA on a positive side of the gel electrophoresis apparatus. So basically DNA, because negatively charged hai, so it moves towards the positively charged electrode, and hence you have to load towards the negatively charged electrode, so that जब turn on होगा, जब वहाँ से current flow होगा, so DNA will move towards the positively charged electrode. So this option is wrong. Option number B, DNA fragments travel along the surface of the gel whose concentration does not affect movement of DNA. Again this option is wrong because concentration does play an important role. Option number C, smaller the size of DNA fragment, larger is the distance it travels through it. This is the correct option because जितना ज़्यादा size छोटा होता है DNA fragment का, उतना ज़्यादा distance वो cover करता है, because वो gel के pores के through move करेगा. So छोटा particle होगा, छोटा fragment होगा, तो ज़्यादा distance वो cover कर पाएगा. Option number D, pure DNA can be visualized directly by exposing to UV radiation. This line is given in the NCERT. Actually this question belongs to biotechnology. If you see that chapter, this line is there that pure DNA cannot be visualized because you have to stain the DNA by staining dyes like ethidium bromide or acridine orange. Without staining the DNA molecule, you cannot visualize that directly in the UV light. So the correct answer will be A, B and D because these options are incorrect. Question number 89. An enzyme catalyzing the removal of nucleotides from ends of DNA is. Again this question belongs to the biotechnology chapter, and it is clearly mentioned that you have to remove the nucleotides from the ends of DNA. So if you see the options, the option number 3rd is the correct option because the exonucleases are the enzyme which cut the DNA molecule from the end part and hence remove the nucleotide from the end part. जबकि जो endonucleases होते हैं, they are cutting the DNA from in between the DNA or within the DNA. So end part के लिए we always use the exonuclease. Question number 90. In RNA interference or RNAi, the genes are silenced using. Again a question from the biotechnology application part. जो RNA इंटरफियरेंस होता है दैट इज़ प्रेजेंट इन ऑल यूकैरियोटिक ऑर्गेनिज्म फॉर देयर सेलुलर डिफेंस. सो बेसिकली हम यहाँ पर यूज़ करते हैं डबल स्ट्रैंडेड RNA को. डबल स्ट्रैंडेड RNA जो mRNA प्रेजेंट होगा उसको साइलेंस कर देगा. सो जब भी जीन इंसर्ट करते हैं तो सेंस एंड एंटी सेंस बोथ पार्ट इज इंसर्टेड एंड हैंस द डबल स्ट्रैंडेड RNA is produced and that double stranded RNA is silencing the mRNA. So in RNAi or RNA interference technique the double stranded RNA is used.